All right, welcome everybody. It's Drug Discovery Diaries podcast, season 2, episode 2. And yes, it's the middle of July, and by the time this comes out, it'll be further into July, and we're only episode 2, and probably thinking, what the heck? But it's been tough. You know, it's tough with some of scheduling in particular, and so it's been, we're trying to work out some of those details. Couple of interesting things coming out in this podcast. First of all, as you can notice, I'm stag today. I'm going it alone. Barry and Hillary are absent today, and so they'll be missed, but they'll be back, that's for sure. I carried them anyway, and so it's not really that important. No, I'm just kidding. They carried me, that's for sure. And the second interesting thing is we're trying video today, and so we're giving this a shot. And I think it's a long time coming. I always wanted, one of my dreams has always been to host a talk show. It's one of those things. I think the podcast came close to that. It was only audio, but I always, you know, one of my favorite Seinfeld episodes is the one where Kramer sets up the Merv Griffin set in his apartment. And ever since I saw that in the '90s, that was one of my dreams. Is I wanted to host a talk show, and so this is the closest I might ever get. And so we're going video this time. We'll see how it comes out. Very excited. I have a very special guest. We're filming from the spectacular Biolab site, again here in the Curtis building between 6th and 7th and Chestnut and Walnut. Outstanding facility, Biolabs for Advanced Therapeutics. The team here is wonderful, and they're always gracious to me and my teams to come and spend some time here. And so we're back here again. It's always a treat. And today I got to go solo. It's bizarre. Very special guest talking about a topic, I guess a platform or a therapeutic modality that we have yet to touch on, and that is gene therapy. You know, I think we've danced around a little bit, but we never actually had a full topic, a full podcast on it. And so I brought in somebody who's been living this for a number of years. He's an old friend, a good friend, and a former colleague, Brian Tomkiwicz. He's a vice president at SK Pharmteco right now. Built up their viral vector manufacturing capabilities. So he's really well connected with all of those details, and we're going to hear about a lot of that. We're going to pick his brain about where the technologies are nowadays. We're going to pick his brain about where he sees applications of the platform, strengths, weaknesses, and the like. So really looking forward to hearing from Brian. As I mentioned, he's an old colleague. We used to, you know, this is the kind of stuff I do. So Brian and I, we first met, was it
13-ish?
13? Was it that? I thought it was earlier than that.
Maybe.
But
12, 13.
he made an impact. He made a dent right away when I met him. He seemed like a brother from another mother because he's got a little bit of an edge. People who know me know the kind of edge I can bring to a conversation. But Brian, he can give it right back, and so I picked up on that right away. He started out. He's going to tell his background.
Actually, yeah, I think we connected in 13 as boss and employee relationship.
Well, that was, but you joined. So why don't you go through it? Tell us your story because long and you know storied past.
So 20 the longest storied past. 2011 joined Janssen under the Synterix group,
right?
So, which is now Aero Therapeutics, which is in the Curtis building here. And then I guess around 2012-ish time frame, 13, I joined up with you.
That's right.
After I was sick.
That's right. Well, but let's go back a little further. But you were educated. You're a Philly boy.
Yeah, Philly boy, born and raised. Went to Catholic school in the city, but then went to public high school. And my career path was kind of interesting when you look back. Like, where could you have been, or where might you have been? I was originally potentially going to West Point. So who
You got the haircut for it, that's for sure.
still do? Yeah.
We'll talk about who you look like in a little bit.
Yeah, West Point or Annapolis, but I think I wasn't really built for engineering, and that's what the academies are.
Right.
Everyone's an engineer at the academies. I was more into medicine, so I didn't take the appointment to West Point. Went to Penn State, and then was also volunteering in a hospital, much like Mike is very familiar with the hospital setting. And then realized I don't really like blood. I was like, so what am I going to do? I mean, I could have been a medical doctor, but after working in the ER, I was like, oh man, I can't.
Wow.
I can't stand this.
And this was in
High school, college.
that's okay. And when the volunteering was in Philly hospitals.
Yeah, it was actually at it was at Tarsville, Frankfurt, but
Okay.
now which is at Jefferson Hospital, up in the Northeast. And so I had signed up to be a candy striper
Just talking about that yesterday with my youngest daughter. Go ahead.
to actually volunteer at the hospital to get in with the doctors, which worked seamless, brilliantly, actually. And then I became sort of friends with the pathologist, and then also volunteered in the ER. And so did a lot of things. I guess the ER docs knew I wanted to be in medical school, so they took me under their wing whenever I would volunteer once a week for about 5 or 6 hours. So it was a really great learning experience.
Yeah.
I advise everybody who wants to be a medical doctor, volunteer in a
Yeah.
hospital and see if you like it first. Because parts of it I love, but my wife's a nurse and she tells me all these funky stories, but I think you're wired for whatever you decide or end up doing in life, and I was not wired to be around smells and bloods and guts. This coming from a guy who will not clean crabs because doesn't like to get his fingers dirty.
You don't clean
No,
crabs?
No.
Well, who does that then?
My wife. And so then I went through college and majored in microbiology, and I had one course in college and I knew exactly what I wanted to do, and I never looked back. And that was a biochemistry and molecular biology 480, so B&B 480, which was taught by Dr. Richard Frisk, who was a JC virus, SV40 virus. And it was basically, the premise of the course was tumor viruses and oncoproteins. So it was essentially viruses that cause cancer. So I was like, well, this is exactly what I want to do. Senior year of high school or college, grad school locked up, got an A in that course. It came pretty easy. It was almost effortless. So I was like, this is exactly where you need to be. And so then I went to Thomas Jefferson, got my PhD in molecular virology, studying herpes viruses, HHV8. Went on to University of Penn for a postdoc under Dr. Ron Coleman, and that whole group with the esteemed Nobel laureate Drew Weissman when he was assistant professor at the time.
Yeah.
Him and his, one underling. And it was going back to early 2000s. So, so I was part of that group and did a lot of HIV pathogenesis. We were doing some work on Lin kinase signaling in an oncology group under the BCR-ABL oncoprotein, which turns out that HIV and how HIV and the macrophages migrate in the brain is almost exactly the same way that stem cells migrate, and ultimately connected to really disparate mechanisms of BCR-ABL induced leukemia versus HIV-induced, macrophage migration. So it was pretty wild. And then that's when I went on to another startup, which was Morphotech, which was antibody development. So because my background was in cell signaling, did a lot of the antibody development work. And then that was from 2006 to 11, and then went over to Janssen for a decade, where I met Mike. Joined up with Mike's team in 2013, and then from there, we sort of, we were pioneers in gene therapy at the time.
They weren't doing it until we got there, that's for sure.
I know we spoke to Carl June, which then became Novartis and Kymriah. Spoke to Catherine High, which then ultimately became Spark and Roche. And then wound up, in 2017, purchasing some assets from Mira over in London.
Yep.
But, all through 2016, 17, 18, 19, we built the whole program over there along with cell therapy and our other good friend, John Lee.
Yeah. He's probably pissed that you're first before
I
him.
deserve to be
but,
first.
But I have to get a bigger camera setup for him because he's a big guy. Can't fit him on the screen.
And I think what messed everything up was COVID. Because everything sort of shut down during that time frame, 2020, I guess 1920, 21. It was hard to get traction. We're trying these new modalities out, for obviously a company like Johnson & Johnson, which is ultra conservative, sort of testing the water, how much manpower is behind this. I guess ultimately it didn't work out. I left the organization in 2021 to do something fundamentally crazy, which was start a CDMO gene therapy, cell therapy, and everything under the sun, Center for Breakthrough Medicines, at the height of what we thought would have been the legendary and epic boom of cell and gene therapy and everything else related to advanced biologics, but that turns out that wasn't the case.
Right.
And then basically learn how to build organizations under immense pressure.
Yeah.
How to lead under immense pressure, how to grow a business, how to create a business, but also how to create name brand recognition. Like who are we? What do we do different that Catalent or Lonza or Thermo can't do? And so just really trying to carve out that niche. And so ultimately we wind up deciding on that, just viral vectors in general. So adenoviruses primarily, AAV and lentiviruses really are what's hot right now.
That's great. Now, I mean, I want to talk about the ups and downs of the field, because I often reference another field that you used to be in as well, and how that went through similar peaks and valleys. But before that, so we overlapped at Jefferson. You graduated when? When did you defend?
Oh, well, I defended in 2002.
Okay.
Well, what part of your
I know.
What part of your 10-year span at Jefferson do we overlap?
All right. No. So the second part, we
part.
definitely overlapped, because I was 98. I guess I defended in 2001. But what building were you in again?
I was in Jeff Hall, and mostly Jeff Hall.
Okay, because I started out in Jeff Hall during my first stint, and then ended up in what was it called, Blumley or something like that?
Yeah.
And I used to have to come to Jeff Hall every once in a while, so we probably were within a stone's throw.
Yeah, tucked in the back of the corner of the, ancestral Jeff Hall building.
I mean, that was the first research building before they built
in the country.
Before they built that 11-story monstrosity across, catty corner from.
Now
there.
Jeff Hall is quite nice, but actually the labs need some attention. But other than that, the rest of it looks pretty good.
That's where I cut my teeth. Something else. And then, so you're part of the alumni board.
Yes, now you are.
And now I am too, thanks to Brian.
Yes. So we're always trying to engage the community, the previous Jefferson graduates, to stay in touch and build a network, and sort of groom the next crop of scientists coming out, which is important.
No, that's good. That's good. I'm looking forward to
U.S.
that.
biotech needs a sort of shot of adrenaline.
Needs a kick in the ass.
Yes, that it does.
It's a kick in the ass.
That it does.
A lot of competition from other countries that have found better ways to sort of get into the clinic. We can maybe touch on that as well. So that's great. No, it's a treat to have you. We got a lot to talk about. Hopefully we can fit it all in. Like you were referencing, when we were at J&J, it was mainly, for the lion's share of its existence, small molecule, and then, they bought Centocor in '99, and so they got into biologics, and they had some great successes there. We were a part of some of that as well. I think that represented, up until a point, the most exciting time for me when I was there. But then, we had an opportunity. So you and I started to work together in 2013. We were still part of the Ancestry Biologics organization, trying to get new targets and programs going using antibodies, which I think we were very successful at. And that was a lot of fun, because up until the point, our only customers from a platform perspective were oncology and immunology. But then we had a chance to talk to, CVM and infectious disease, and even the neuroscience department, which was interesting
Yes.
to see how to apply them. But then we had the opportunity to build up something completely new under Bill Stroll, which was cell and gene therapy, or as our boss liked to refer to it as gene modified cell therapy. I can't remember why he was adamant about using that terminology, but in any event, the premise was, antibodies. These are the words I use. Antibodies are played because everybody was doing them then, and everybody was kind of doing the same thing, blocking a cytokine or whatever. And there were some, your rituximabs or whatever that were successful, but everybody was doing it. Like Bill used to use the word commoditized. I mean, literally, didn't need to have an organization to make it an antibody drug at the day. And so he was always a visionary and always thinking about what's next. And he saw the writing on the wall with regards to what was coming out of Penn, with the cell therapy stuff, with the gene-modified cell therapy and the CD19 CARs. But also he thought, and maybe I had something to do with convincing. I was always a fan of gene therapy. I thought there were opportunities there to apply those technologies, which were, look, it's classic, that was something talked about in the 90s over and over again. Once, people started to figure out the genetic basis of disease and those kinds of things, replacing defective genes with new ones. And it had some tough times in the 90s, some mortality, some deaths. And so it sort of hit the bottom. But then there was a resurgence because people started to find ways to make those vectors safer and understanding the mechanisms of what caused the fatalities. And then in particular, AAV, which was just getting hot, I guess, in the mid 2010s.
Yes.
Jim Wilson was bringing it back. It's a virus that was, I guess, you know this better than I do. That was like hidden in a culture of cells making adenovirus.
Yeah, that was contamination.
Contamination, and it is. You can talk about the details of it, but it really got things going again. And in point of fact, I mean, after we got our first cell therapy opportunity up and running, the second thing we got going at J&J was a partnership with Penn's gene therapy. I forget what they called it, but they're
GTP.
Was it GTP?
Yeah.
And with a strategy to use AAV to deliver a biologic for influenza, a broad spectrum biologic that inhibited multiple strains of influenza. And so that we got that off the ground. I mean, that got pretty far. I mean, it got to enemy declaration, but then I left, and I don't know whatever happened to it after the fact.
Then IDV closed down,
Then yes, oh, that's right.
then
Yes.
CVM closed down.
Yeah, so there was lots of changes happening around that time period, 2018, 2019, 2020 in particular. But I think we both, and you were excited. I think one of the things we started to do together when we were working together was trying to build the capabilities to understand and use AAV internally, because I think, although you can send everything you want out to get made, I think to truly understand your drug product, you got to do a little bit of internal work, and you built those capabilities.
Yeah, exactly.
I threw that in your hands, and literally you ran with it. And
I mean, the beauty of it was that, obviously drug companies want therapeutics for profit, but we were using AAV at Janssen at the time as a tool reagent.
Yeah.
And really doing a lot of work with the disease area strongholds, or the DOSs, to help build reagents for them.
Right.
So how do you overexpress or underexpress a sort of a protein in an animal or in a cell? Well, it's easy just to deliver a viral vector and mimic a model.
Yeah.
And so we were, designing all kinds of stuff, and then I was doing more funky things, like taking some of our pretty wild antibody constructs, either bispecifics or even trispecifics, packaging them into an AAV and saying, hey, well, we can do systemic delivery with sustained expression. So, like, they were the kind of things we were building while we're looking at, opportunities for acquisition. So I guess in 2019 timeframe, cardiovascular DOS built out the retinal
Right.
DOS, or the cardiovascular TA therapeutic area built the retinal DOS, and so we were then going to start getting into the retinal diseases, which are, for AAV gene therapy is low hanging fruit because it's low dose and it's easy to administer.
Right.
So that lasted not too long.
Right.
Remember, we hired somebody, and then a month later they were no longer there, and I never really knew what happened. And so, one of the things we needed was the buy-in from the TAs to support the programs, because ultimately they're the ones who were
Funding everything
and pushing the drugs into the clinic and declaring NMEs, and so some of our champions that were doing that work, and especially neuroscience TA, then subsequently took other positions elsewhere in basically established gene therapy companies. So we're kind of left on our hands and wondering what the heck's next. And our manufacturing prowess at J&J was not in gene therapy; it was
Right.
in the antibody world. So
Right.
I was kind of leading that charge, basically having learned everything what was self-taught.
Right.
And you think about gene therapy in general, this antibody's been around for now going on 50 years, I guess. Yeah, 50 plus years, where production systems have been well validated. But gene therapy, like Mike said, it really didn't start until early 2000s. So back in 2017, which was almost 10 years ago, recombinant AAV platforms were only about a decade old.
Right.
We didn't even have suspension culture.
Right.
Everyone nowadays, we're doing pretty well in that space, but back then everything was adherent culture.
Right.
And so this production gap, which was effectively termed this loss in titer from adherent to suspension. So everyone wanted to do like this let's grow cells on an adherent and then just put them in a bioreactor for a couple of days.
Right.
And that's called our suspension culture. Well, obviously that doesn't work nowadays.
I mean, one of the issues I think with AV as a platform in particular was always manufacturing costs. It's like,
still is.
It's one of those. It's an incredible platform when you think about it, and you get to know about it. I mean, maybe you can tell us some more details about it. But a huge thing in drug development is always, well, how much is it going to cost to make?
Exactly.
I mean, even in the antibody world, I love to bring up this example of the antibody world. When I got started, in the early to mid 90s with Centocor, they were using myeloma cells and ascites that we asked, maybe that was before that, and then they moved to myeloma cells, and they were fed batch cultures or what.
Yep.
Like, it progressed, and it was this gradual progression where
the recombinant platforms
where you were hundreds of milligrams per liter to single digit grams per liter to what it is now. I don't know, 10, 20 grams per liter.
Yeah, I mean, I guess
CHO cells
12 gram per liter is a pretty good platform.
Yeah, so it's always I think.
And we're getting there with AEV. There's no doubt. I mean, you look back over early production systems using adenovirus primarily until sort of Jude Samolsky built, I guess, the first recombinant platform in the late 90s and showed that you could, this P helper concept, take the helper genes from adeno, put them on a plasmid, and use this triple transfection method, which is still used today.
Right.
Maybe E9, E10 was a good titer, but now, a good platform is pushing E12.
Okay.
So we're 100 times better than what we were.
Yeah, no,
which is a lot.
No, it is. I mean, when you think about antibodies, that's the type of productivity that is the ultimate goal. But so let's get back to basics. So AAV, so adeno-associated virus. Tell us about what it is, why it's considered a great platform.
I mean, so yeah, AAV
stable.
In general,
does it integrate? Does it not integrate?
From when you think about, I mean, the risk has always been that once you deliver this virus containing the payload, that's it. You open Pandora's box. You can't undo it. And so whatever is delivered is there in perpetuity. So there's always that sort of the conservative risk that what do we do if it gets out of control, or how do we put the genie back in the bottle? I mean, obviously we're talking AAV resides in the monogenic disease space, so we're targeting, it's still primary function is how do we target cells that lack a gene.
Right.
So in that respect, overexpressing a human protein in a human is not going to really cause too many problems. And we can control dosing. There's ways to control dosing, but the amount of virus, inducible promoters, tissue tropism, stuff like that. So, I mean, the risk is not inherently far-fetched. The virus itself, AAV, is really not a harmful virus. If you had wild-type AAV, it would only replicate in the presence of an adenovirus because you need the adeno helper proteins E1A and B to actually replicate AAV. But in and of itself, AAV is not a cytopathic virus. It doesn't really cause it doesn't cause any human disease. So in that respect, it's a great vehicle to deliver a payload. There's rep-associated toxicity. The virus doesn't integrate, it does maintain episomal, so that's also good. So you're not affecting the genomic structure of cells, so you're not necessarily integrating behind an oncogene and causing cancer. Now there have been some reports of cancer, but the incidence of cancer in some of those patients were high dose, but also had those individuals did come with some comorbidity. So whether the virus caused the cancer or something else remains to be decided. But when you think about it, it's a very simple virus. It's a hardy virus, so you can engineer it quite effectively. So you have a limited payload capacity, so that limits your usable footprint space.
And it's literally the AAV ITRs.
The ITRs, which are required for packaging,
and that
your
stands
GOI,
for
the inverted terminal
repeats.
Repeats.
And so those flank your
Gene of
cargo of interest. So
That's it.
usually that's a promoter of some sort that works in a cell type you think is going to be
Exactly.
transduced, and then your
And that's
gene
it.
of interest and a stop signal or something
And
like
that's
that.
it.
And that's it. That's, I think, one of the
That's the simplicity of the whole thing.
It's unbelievable. I mean, I think that was what attracted me to it. The other thing that attracted me to it was it's not enveloped.
No,
So
it's not
it's literally a protein shell around
That's it.
DNA cargo. It's like, I love that. You know what I mean?
And so that's what makes. That's the beauty of the whole platform.
There's issues with that to some extent because that protein shell is foreign, and we'll talk about immunogenicity and that kind of stuff in a little bit. But the simplicity was breathtaking.
Yeah, when you think about trying to produce something in a recombinant system, having a virus that essentially can withstand pure hydroxide and pure acid and still retain its structure and still be infectious. As opposed to say a lentivirus where high salt or detergent or outside of normal pH will destroy it instantly. Time, temperature, all those things. So an AAV virus really is indestructible. So, of course, what you always like to have is specificity in some respect. We can control specificity, like I said, by tissue-specific promoters, but if you're trying to target an eye, or you're trying to maybe target even something systemic in the body, subretinal injection is one thing that you can get on target delivery very effectively, but what if you're trying to go systemic? The dosing requirements are high, but how do you get it to the actual cell you want? That's always the problem with AAV, is that most of it will go to the liver, and so, you're trying to target, a heart cell,
Right.
or, a cardiomyocyte or something like that, and 10% of your effective therapy is actually going to where you need it. So, there are ways now, the field's evolving into engineered capsids and targeted capsids, but still, they're very expensive business propositions.
Right.
Because it's all translational. You can only build the best animal model and hope that it works in human.
Right.
But those are luxury items, and so small startups can't really afford elaborate capsid, cloning and capsid engineering strategies. So, it's one way to reduce immunogenicity, lower your vector burden, as well as get on target.
Right.
Three things you want, which then ultimately lowers your COGS because then you don't have to have massive scale.
Right. Have they made advancements with targeting?
Sure. Absolutely.
There used to be this talk, there are the different serotypes that had a predisposition to this tissue type or that tissue type. I don't know if that.
Yeah, we take advantage of some of the natural tropism of the viruses, AAV9 versus an AAV1 versus ubiquitous AAV2, but they still do go to the liver and they still go fairly everywhere. But you can skew it a little bit in your favor. But yeah, there's lots of companies have spun out of novel capsid engineering. Aperture is, doing pretty cool stuff with blood-brain barrier, permanent
Right.
tropism using transferrin receptor, et cetera. So, and there's others that have, 4D,
Yeah.
Dino, which was doing complete AI and AI targeting strategy. Again, at the time, we spoke to Dino when they were sort of in their inception. It was kind of wild when you think like, Dino was proposing we can make, a billion different types of capsids. I'm thinking, well, how do you screen all these?
Right.
But they've, done it remarkably well in this space. And so, one of the original 4D was one of the original companies that was doing capsid evolution.
Yep.
So the field has come a long way. There are other boutique-ish ones, some that have been consolidated into Voyager, doing some pretty cool technology.
Yeah.
So, but yeah, the field is definitely heading that direction because when we get to, as the low-hanging fruit tree dries up, you got to go more bigger and bolder, which is systemic disease. And
Right.
systemic disease requires, obviously, you can't have pre-exposure.
Right.
So a classic AAV 1 or 2 or 9 may not work. So
Right.
you got to find something unique and novel, as well as trying to reduce immunogenicity. So how do you do that? You have to use a capsid the body's never seen before.
Right.
But then that also brings into manufacturing problems. It's one thing to have a vector that can go where you want it, but can you make it?
Right.
And so there's a lot of considerations around highly supple capsids and how you manufacture that, because
Right.
it's not as straightforward as thinking it's going to behave like AAV9.
So I think there have been. I mean, is there something approved? I mean, with SMA or these muscle wasting diseases?
Yeah, SMA is AAV9.
That's systemic, right? Or
Yes,
is it
that's systemic.
multiple local injections?
Yeah, I mean, obviously trying to get it into children because they weigh less, so
Right.
there's a lot. But still, I mean, at the time, I believe that each manufacturing run was per patient,
Right.
and so when you think about the cost of that, GMP manufacturing run to produce a batch for a patient and
Right.
all of the quality that goes along with it, it's an expensive proposition. That's
Yeah.
why we're talking multi-million-dollar therapies. But then the actuaries will look at it over the cost of the therapy being a certain amount, and it's cheaper for the gene therapy.
I think it's still probably a platform that is going to stay exclusive for rare diseases or gene replacement, in which I think this is one of the selling points over the years where it's been thought of. It has a role because these people are going to die.
Absolutely.
Or they're going to be on expensive drugs for the rest of their lives anyway, and I think that's part of the way it's justified, the cost. It's like, look, one dose may cost this much money per patient, millions of dollars or whatever it is, but you don't have to give them something for the rest of their lives, and that would tally up to the same thing, not to mention having to be on. So I think, but it's probably going to stay in that niche.
Yeah, I mean, rare diseases is where it's at for gene therapy. When there are no cures, but there are known mechanisms of how these diseases, and they're usually fairly straightforward.
Right.
And the key to is early intervention, right? Because gene therapy doesn't repair disease. It can stop progression of disease, but once damage is done, it's very hard to undo that damage.
Right.
And so a lot of the hereditary brain diseases and other types of monogenic diseases where it resides, but obviously, and we see this a lot at the commercial manufacturing, we have either advocacy groups or even parents raising money
Right.
for us to manufacture a drug for them.
Right.
And so the industry, us as innovators and drug developers can only do so much. I mean, the industry as a whole has to sort of maybe change their mindset. I obviously understand why drug companies look for the next drug or blockbuster that can net them a couple $1 billion to continue the cycle of continue to develop drugs, but where you might only have 50 or 100 patients.
Right. The concept of blockbuster.
It's
It's tough to see that translated into blockbuster.
It's not going to, and that's where rare diseases really need the help. Where maybe all the drug companies pay into a pot to allow these technologies that there's access, because really they can be cured, but it just, and of course the regulatory agencies have to do their part as well. And I think they're loosening the grip a little bit with the concept of, disease X and disease Y and disease Z using the same vector. Okay, well, guess what? You can use your CMC package
Right,
across all platforms. That's a massive, massive reduction in regulatory constraint. And that the regulatory cost and showing like for like or comparability and being able to do it in a platform perspective is huge, because now you can expedite and really save on costs, and you don't have to continue to reinvent the wheel every time you do something. So that's where it's going to start. But obviously, the rare disease space and the things we're doing in rare diseases and trying to make it affordable. How do you do that? By production and producer platforms where you don't have to rely on expensive plasmids.
Right.
Using very high expression cell lines, very good downstream and upstream, very good analytics, understand what you're making. All those types of things all contribute. Now, we do move into systemic diseases, and eventually maybe one day we'll get to a spot where, something like AMD, where you just get, and people are doing this where you can use an AdV to deliver an antibody and have systemic expression of an anti-VEGF or something.
Yeah.
So those are the types of things eventually you could get to.
Right.
So instead of repeat dosing of an antibody, you just give single administration of an AdV. So
Interesting. And then the toxicities that have been seen, they're usually the result of just the viral load, right?
Well, yeah, I mean, water's toxic.
Right. Good point.
Same thing with a virus. I mean, water's toxic. You drink too much of it, it'll kill you. Same with a virus. I mean, we're giving these whopping payloads, E13, E14 vector grams per kilogram. We're on the order of 10 to the 17th or, vector, 10 to the 18th vector per patient. You have, it's a virus. They will elicit immune responses. You will have residual protein around, no matter how good your system. There's still residual protein. There's still some residual DNA. You just can't get rid of. Maybe a low level endotoxin. These are biological systems, so they're going to have trace amounts, but we try and get them down to as low as possible. But when you give that much of something, you're just overloading the system. So, I mean, I know that now people have sort of co-administered immune suppression therapy, or steroids or local steroids to control that reaction. But I mean, these are infusion reactions that happen over a couple hours, but still, that's a whopping vector load.
Got it. Now, like we talked about, the AV does not integrate, and in principle, if it transduces a cell that does not replicate, it'll stick around for the lifetime of that cell.
Well, that's the beauty also of AAV, right? So post-mitotic cells will still get the virus. So a muscle cell is a prime example where it'll just hang out and crank out protein. So, unlike other viruses which require cell cycle or active cell replication, the AAV does not. So,
Now, the flip side of that are the integrating viruses like lentivirus.
Exactly.
And that is still, although that that is still the primary gene delivery vehicle for ex vivo modified cell therapies, so your CAR Ts. And that integrates somewhat randomly and is the go-to system for that. It's got competition now because people are looking at in vivo engineering using lentivirus as well, pseudotyped, but also nanoparticles. Where do you see? The in vivo is a gigantic, step forward with regards to lowering costs.
Absolutely.
And the like, with regards to that modality, engineered cell therapies, T cells in particular. But do you see, any insight into where you think that might go? Because you're definitely in the lentiviral space. That's another thing
the lentivirus. So that's actually the hottest thing right now is in vivo lenti. When you see these things that just catch fire, and you're like holding it.
Right.
Shameless plug for our company. We're probably one of the leading. No, we are leading in LVV purification methodology, and we're We have some patent process as well as a publication coming out soon. But really, we've enabled in vivo lenti to sort of flourish because one of the major problems with lentivirus in general is they're so damn difficult to work with, right? Because they're so sensitive.
Right.
They're lipids. They have a lipid based off the plasma membrane. Shear force can just strip proteins off. So there's no affinity resin like an AEV where you can get super high, take very large volumes, run it over an affinity column and get it down to a small manageable volume where you can do high salt or low salt or high pH or low pH. So you can take advantage of extremes and get rid of impurities. You can't do that with lentiviruses because it will absolutely destroy the virus. So we've sort of really, and we have a lot of white papers on this field and we've presented at ASGCT, but we've been able to get our residuals and the yields up, very high with almost, non-existent or undetectable residuals. And so the quality of that virus to be able to go into an in vivo setting is amenable, right? So it's one thing you have to keep in mind osmolarity. You have to keep in mind that what you're putting to a patient, you can't harm the patient while you're trying to deliver therapy. And so we've enabled that. But what we've seen, ex vivo primarily being the cell therapy piece, i.e. we take a virus, we kind of don't care what it looks like because we're going to put it into a large bag, we're going to infuse T cells, we're going to select those T cells, expand those T cells and put them back into the body. That's the classic methodology. That's obviously expensive, time consuming, a lot of cold chain considerations to how do you store cells, freeze cells down, expand cells, how do you ship all that. And so that's only accessible at very large hospital centers.
Right.
So patients have to come to a place like Penn in the city here or Jefferson and can only receive treatment.
Right.
It doesn't help somebody who doesn't have a lot of money in the somewhereville United States. So if you eliminate the middleman, which in the case is the cells, and you can just efficiently deliver the virus to the target cell, i.e. for the most part we're talking about liquid tumors, and do it well enough to not cause other issues to the patient, you've just eliminated a month's worth of work and everything. So we're seeing it now. I mean, if you look at the field in general, the results that come out almost every day from all these companies, Colonia leading the charge with every evaluation. If you look back to it, one of the first, which was in Diaries Bio, now we're talking, $500 million increment for every next in vivo CAR, right? So now we're up to $3.5 billion. The next one's going to be $4 billion, and it's just staggering to see this. But the results we're seeing are just unbelievable.
And so that as far as the technology for pseudotyping to get it to specifically interact with T cells is pretty specific.
Yeah, well, no, it's actually pretty cool because it combines our world, man, right? So you think about a virus, so the VSVG goes to the liver, right? And it targets everything to the liver. And so you can reduce those capabilities somewhat by eliminating LDL receptor binding, which is primarily in the lipoprotein density receptor. But how do you then get it to go to T cells? So we can strip away the binding, but so here's where it combines technology, which is CD3 redirectors or VHHs or stuff that we were developing at Janssen, which is a lot of the premise behind a lot of what bispecifics are when you think about it. So, you can take something that really has no purpose because a drug developer may not want it as a therapeutic because it doesn't meet their TPP, right? So maybe it doesn't have high enough affinity for a therapeutic. But hey, we don't care about affinity because we have avidity. Right. And we just need to stick it on the surface to make it go to wherever we want it. So you're seeing a lot of that technology now with these redirected viruses. Right. And whether they're fusion proteins of VSVG or they're 5 plasmid-based systems where, or you take away the integrase, which is another way to improve safety, where we just, basically the virus is a limited lifespan. So it delivers payload, doesn't integrate because it lacks the integrase, but it can deliver the payload enough to make the CAR do its thing, and that's it. It just disappears over time. So the safety, switches are built into these things, and they're working unbelievably well.
I forgot about the integration or integrase deficient versions of lentivirus. Is that where the field is going, you think?
Yeah,
because one of the things I always, sorry to interrupt, one of the things...
Well, you don't want to integrate behind an oncoprotein, right?
Well, I mean, so there's that, and then there's also, one of the things that I loved about AAV, and we talked about already, was did not integrate, and obviously you would use it in situations in which you know, you were targeting cells that were post-mitotic, like you said. The other the targeting cells with lentivirus, you know, re-pseudotyping them is a great thing that it integrates. But I always had an issue with the fact that the CAR T that comes about after the fact, it's around. It ain't going away if the virus integrates. And so this integrates deficient is because the other way is like a nanoparticle.
Yeah, the LNPs.
That's also now.
Sure.
I mean, do you have an opinion about
No,
integrase deficient versus LNPs? Because LNPs bring a toxicity
Yeah,
with them
the
as well.
LNPs are inherently. I mean, nothing's easy, right? And I always think it's sort of where you're born and raised and how you're raised.
Northeast, South Philly.
Yeah, there's no right or wrong answer to it. It's just what you're used to. And so people who are raised in an LVV world are comfortable with that system, and people who start out in an LNP, you know, LNPs are not magical beans.
Right.
They're inherently immunogenic. They're very hard to manufacture. Composition of matter in LNP, controlling composition of matter, encapsulating RNA, basically RNA, the quality of RNA.
Right.
I mean, there's a lot of consideration. So it's not turnkey by any means. And I think a lot of times it's accessibility. It's much easier to order a plasmid-based system and make a virus than it is to figure out how we're going to set up an LNP manufacturing platform. Right. And when you think about it from a biomanufacturing perspective, large-scale production, it's very challenging as well. CMC is very difficult. I mean, you're dealing with highly volatile organics in very large quantities.
Okay.
And then controlling all those reactions. Cholesterol is very important, right? You can't have too much cholesterol in an LNP because you can't administer cholesterol to people.
Right.
And so how do you control those reactions? And then, of course, you get into the third part of that, which is targeting LNPs. You know, how do you take advantage of LNP targeting? Where in an LNP, everything's going to the liver, literally everything, because they're lipids. So do you take advantage of different tropism of lipids for specific tissues, or do you then introduce now targeting moieties?
Right.
But yeah, at the back end, if you can figure all that out,
right.
if that's a big two-letter word, you have the advantage of an RNA delivery system, which of course you also have to consider RNA can be quite immunogenic as well.
Right.
But it's limited. It does its thing and moves on. Right. So I think the virus does a very good job of shielding the immune response, getting, that because of the way the virus unpackages and delivers RNA behind the sort of TLR firewall to allow it to do its thing, whereas naked RNA delivery via an LNP, where it just endosome just sort of dissolves and spits the RNA. Well, that's immunogenic.
Right.
On the forward-facing side of the TLRs and stuff. So, I mean, I don't have any skin in the game as far as advocating one platform. We don't do LNPs, but we're very good at LVV. It's just a whole different supply chain constraint.
Okay. But as far as to make a integrase deficient lentivirus, it's just a different vector setup when in the manufacturing process.
Sure.
Right. You just delete it.
Yeah. Exactly.
Right? And that's pretty much standard practice nowadays. That's not what Colonia is using.
There's no standard practice. I don't comment on individual programs, but everyone has their own unique flavor.
Okay.
Yeah. So whatever formula works.
Yeah. Okay. Fair enough.
Much. And I think one of the good things now we're going to start to see pretty soon, and our group's going after it, is solid tumors, right? So CAR T's never really, you couldn't get a CAR T to a solid tumor. Mm-hmm. But now that we're able to sort of take advantage of the trickery involved, we can start to hit these solid tumors, which will open up the field.
What do you mean by trickery?
Molecular trickery.
Okay.
Getting things on target, taking advantage of the antibody world.
Okay. Gotcha.
Because again,
always comes back to antibodies.
Always come back to something. Yeah. VHHs and VHOs and stuff where you can just stud the virus particle with these targeting moieties and basically use it as a warhead to deliver payload. And then you get this sustained expression in solid tumor. So, I mean, they're invaluable. if you look at it through different optics, the way I like to, you and I like to look at it,
Yeah.
and you start leveraging innovating technologies, you can really do some powerful things.
One thing we haven't talked about is standard adenovirus.
Oh, adeno, yeah, they're making a comeback.
What are they? How are they being used? And they're that's also non-integrating, correct?
Uh, no, adeno doesn't integrate.
It does not.
No,
But it is enveloped, right? Or is it not?
it's an envelope. I mean, it's a protein.
It's like AAV, but a little more complicated.
A little bit, a lot of bit more complicated. I mean, AEV being in total 4.7 kb from ITR to ITR. Now we're dealing with 36 kb payloads.
So what the benefit of choosing adenovirus is just payload for the most part.
Yeah, well,
Potential.
no,
Payload potential.
obviously. So for adeno, when you think about classic gene therapy, probably not going to see adenoviral delivery. It's a wickedly immunogenic virus, so it's going to elicit a massive immune response. But we're seeing a resurgence in anti-tumor where you actually want an immune response.
Okay.
And so, like the concept of oncolytic viruses,
Right.
using adeno. Adenoviruses, it's an old technology, 1950s technology. Hasn't really done much in almost 100 years. And there are no licensed therapies in the United States for adeno.
Okay.
There were 3, 2 of which went over to China. I don't even think they're using it anymore. One of them was Ono Therapeutics, which was then bought by GSK, which was then subsequently, I believe, shelved.
Okay.
So there's no licensed therapies for adeno. But, we're starting to see a lot of resurgence in the cancer space.
Okay.
I guess there's, the old school platforms they're not efficient. So one of the products that come out of that is this replication common adenoviruses, because they essentially use the E1A protein of 293 to complement the E1A deleted version of the virus. But there's recombination, so the Percy-6 cell line that Crucell had made at Janssen Cell Company was actually solved that problem by using a truncated version of E1A. So really there was no problem with replication competent adeno. But you obviously don't want to give a therapy that's going to have a virus that's going to actively replicate.
Right.
That's going to cause problems. It's going to lead to cancers, adenocarcinomas. Newer generation platforms now have two vector system where they essentially put all the helper genes required to make a virus in one virus, and then they have this stuffer plasmid that has your GOI on another virus, and you kind of mix them together, and the helper virus can't replicate or integrate its payload, i.e. the structural proteins, and the only thing that gets packaged is the GOI, much like an AAV. And so that's reduces safety, almost eliminates the risk increases safety exponentially. So we see a lot of new therapies coming out now in that space. Again, it's a vector system, i.e. we just transduce cells with virus. So it's, there's some considerations in GMP where you have to master viral banks, but a little bit, I'd say, cheaper than making large-scale plasmid banks.
Okay. GMP.
So still not a lot as far as, the field is dominated by AAV and LVV, period. Okay. 99.8% of the share is those two viruses, and 0.2 is adeno.
Okay.
And that goes back to where you grew up as well. Having access to adenovirus, they're not things you just buy.
Right.
The sequences, they're hard to find. The capsid sequences, the capsid structure, or the pentons and fibers are very complicated structures.
Right.
And so engineering that is not easy.
Okay.
And so they're built out of the old school labs that universities that sort of have this legacy knowledge.
Right.
And then subsequently spin out technology, but it's not like you can go on a website and say I need an AAV plasmid, and in a week, a couple of days later, you have a whole production system at your fingertips. So that's sort of the accessibility which has limited adeno.
Okay, interesting. No, I think a nice segue to one of the points I wanted to make, which you mentioned how cell and gene therapy, the ramp up in the late 2010s, and then the sharp drop off, 2020 and onward, it like I always like to use this reference. I mean, antibodies were that way. I mean, antibodies, they go back to the late 80s, early 90s, and everyone were calling them magic bullets. They're going to cure every disease in the world, going to be spectacular. And then, the first ones were fully mouse, and they did some damage. And
Yes.
so then they were in the crapper. And so they hung out for a while until people figured out, well, we can make chimeric antibodies, and then we can eventually make full human antibodies. And,
Exactly.
and then the manufacturing pieces followed, improvements there or whatever. And then, in the late 90s up through most of the early 2000s, the heyday where, every company in the world was making antibodies, and they were bringing in tons of money and doing patients a lot of good. I don't doubt that cell and gene therapy.
No,
I mean, that maybe they're all ready.
No,
with,
we're good.
with
We're there.
The in vivo, delivery.
The production systems are what. I mean, that's what makes antibiotics. I mean, obviously, antibodies are not toxic to the host cell when you make them, and that's key. You can produce.
Right.
Let's say a lentivirus. There's only so much virus a cell can crank out. Right.
Right.
It's taking pieces of the membrane with it every time.
Right.
So each cell is limited. Now you got to be able to pack a lot of cells in and take advantage of the footprint, which is in process identification of perfusion systems. But even at that, to try and, if you think about it, it's all when you look at manufacturing a virus, it's all quite simple, but it's all very complex because there's a lot of unused space that we just cannot take advantage of. So you think about a bioreactor in a perfusion setting for antibody continuous culture.
Right.
Just feed the thing and it cranks out product.
Right.
It's not that easy in a viral world because even if you could, first of all, if you could have a fully integrated production system, eventually the virus is going to kill the cell.
Right.
So the rep protein, you have rep-induced toxicity if it's related to AEV. You'll have other related, all the E proteins of adeno.
Right.
And then of course you have all the toxicity associated with VSVG and REV as well as the envelope budding and LVV. So you want to maybe get intensification of trying to pack in a whole lot of cells into one area.
Right.
And take advantage of maybe extend it, but you're not going to get like a 2 or 3 week process.
Right.
You might get a 5-day process and that's it.
Right.
We transfect cells at 1 to 4 million cells.
Right.
When you can get up to 40 million. So you're only using 10% of the usable bioreactor footprint or the media availability. You couldn't transfect 40 million cells with 3 plasmids, and
Right.
it's too expensive. There's too much to pack in. So in that respect, we're just doing these sort of what we call just transient production systems.
Okay.
Now we tried to build platforms where all the variables have been eliminated such that you're getting maximal production every time. But literally every time we set out to make something, it's not like a campaign. It's like
Right.
run it, 2 days later harvest, DSP, file,
Right.
and then we repeat.
Okay.
So that's where we are. The packaging systems obviously can do some good in that because we can eliminate plasmid costs. We can get a little bit higher titer because now every cell is making it. Triple transfection it's a think about the efficiency of every plasmid you try and add to the system decreases exponentially.
Right.
Your efficiency. So you're not getting 80 to 90% of the cell. So of the 2 million per mil that we're working with, we're probably only transfecting less than 500,000.
Okay.
Just because of trying to get three plasmids in one cell. So it's highly inefficient. And then on top of that inefficiency, we have to deal with what I see a lot of is just bad engineering of plasmids.
Okay.
So either the GOIs are not optimized so that you get good packaging. There may be other issues, replication competent, safety concerns you have to contend with. So when you start re-engineering plasmids, you can affect the titers.
Empty particles, is that still an
empty
issue?
particle, absolutely. I mean, separation of empty particles. Those empty particles do nothing except improve the immune response.
Yeah.
In a negative way, right? You're just augmenting the immune response with just naked particles that do nothing. So,
and there are ways to.
yeah, we can do pretty well in the AEV space, but still, it's challenging, right?
But even so going back to the gene of interest and that kind of stuff, the payload, there are there's knowledge nowadays about sequence variants that package better than others?
Yeah, I mean, we can see, so heterologous promoters or viral specific promoters that drive high expression transgenes, where you have complexity in a gene of interest sequence, whether it be hairpin structures or high GC
Okay.
or things that are just not liked by cells can be spliced out.
Okay.
And so you start splicing promoters, you're going to lose activity,
Okay.
expression, and they will cause the variability you see. Over packaging, how do you do over packaging, whether it be two vector systems or, every time you increase the size, you risk, truncations or
Okay.
splice variants. And so understanding, I always like to deploy long read sequencing very early in process to understand, you can do long read sequencing on a shake flask product to understand what's the intactness of my transgene before I go into full GMP.
Right.
And then one of my release assays is pack bio, and then all of a sudden I see truncations in a promoter, which
Wow.
could have been avoided early.
Yeah.
So, again, we go back 10 years, NGS technology was not accessible.
Right.
So people never really did viral sequencing. Now it's easy. It's
Okay.
cheap, it's easy, it's fast. And so I deploy PacBio or NGS, MySeq, any of those assays everywhere along the way. Every plasmid we do at SK is NGS sequenced before it even goes into a.
Okay.
So we're maintaining ITR integrity to make sure we have the actual sequence. And then we like to NGS the viruses. Okay, what does it look like? Are we still seeing that same package sequence, or do we see other issues?
Okay. I mean, we could talk about this forever because
We could talk about it forever.
The complexity of these systems. I mean, I think one of the beauty, the complexity brings great concepts, or the uses of them and the utility of them. Is a benefit of that complexity, but everything you just talked about, we're barely skimming the surface of how to. That's why I think it's still the early days. It reminds me of ascites for antibodies.
Well,
or,
I mean,
pre chimerization type of things where it's just on the cusp of making a breakthrough.
The vast majority of failures in gene therapy are in CMC. And so CMC is basically manufacturing. I mean, if you think about this very simply, we do the same things in it. So antibody is a protein. There's no payload.
Right.
Virus is a protein, but we have a payload.
Right.
So two things to contend with. So what do we do in the antibody world? Well, we deploy mass spec like it's
right.
Going to the candy store. And in our world, it seems to still be a luxury item.
Right.
It's not. It's a nice to have, but not a must have. But mass spec NGS are two assays that I push every process step, everywhere along the way. Why? Because one, mass spec controls PTMs of your capsid. What's happening to the virus as we manufacture it, as we stress it through, whether it be TFF or we put pressure on the virus. We induce salt. We put pH changing.
Right.
What's that doing to the capsid? And it's doing things to the capsid. Okay, what's happening to the payload? Remember, these things are going into the virus.
Right.
It's not a solid ball. There are pores.
Right.
Is that negatively impacting? So if you look at those two assays and just use them constantly through your process, you're always going to know where your failures are. And then one thing we don't talk about, which is formulation.
Right.
You can't build your whole R&D strategy and all your tox models and animal studies with a virus produced this way, and then all of a sudden, okay, guys, we have a great package. Now we put it in manufacturing, and everything changes.
Right.
Or we don't take into account formulation until it's like we have to formulate, i.e., GMP product because it's going to humans. No, they are one thing.
Right.
They have to be thought of as one thing, and one platform, one everything, so that you have that comparability package. Because you don't want to get in a situation where, our R&D team makes it one way, but our PD team and our GMP team makes it another way. Well, now you just introduced a massive amount of variability.
Right.
What am I to believe now? Do I have to repeat all my assays? So we always like to just do one approach where everything's the same. It's the same cell line. It's the same plasma. It's the same columns, the same everything, and same formulation, right? So at the end, that product, is identical.
Right.
And so you have that translatability package that can last with you. It makes your BLAs a lot easier to interpret as well. Because one thing you don't want to do is get to the end, you're ready to file, and then, oh, by the way, our plasmid's messed up, or, oh, by the way, we're getting, massive splice variants, and the agency are going to say, how do you go fix it?
Right. That's right. That's right.
Well, there's
Spend more money.
you could have avoided early on just
Right.
by running. No, all the major CROs will do NGS. It's very cheap. You
Okay.
just run it at least once. It's something you have to do all the time. Once you have your process locked, nothing really changes, but you at least should look early
Right.
and say, okay, look, we're not doing anything bad. So, and then periodically, obviously we check it at the end, but every time we do a batch, large scale production of plasmids, we NGS it.
Right.
Because you never want to introduce mutation and then, oh, by the way, realize
Yeah.
we have a problem. We just spent $2 million on a plasmid.
Houston, we have a problem.
Yeah, so that's the CMC, and that's what, often overlooked.
Well, this has been awesome. I mean, like I said, we could probably talk about this for hours and hours and hours, and I probably think we will.
It's not
in
about
the
us.
future.
4 folks on the camera have to listen to us.
Well, but anyways, I mean, so it was great. Thank you for taking the time. This is, we're pioneering a new medium, which I think I'm excited to see how this works. Obviously, Brian, a spectacular guest. He likes to think he looks like John Cena.
Yeah.
I think he looks like Bart Simpson.
The old Bart
Simpson. So that's just something between us. But no, thank you very much. Looking forward to following up. Maybe we can try this again in the near future. Get the other 2 clowns in here to see what they have to say. Because Barry's spins more or leans more towards the manufacturing side. So you guys could obviously go off, and Hillary on the preclinical side. I'm sure she'd have a lot of things to say there, especially approaching INDs and the like. So
Yeah, I mean, one of the topics we didn't talk about, which is so difficult even in gene therapy space, is the potency assays. How do you create, because the viruses work unbelievably well in people, but they do not work in vitro.
Right.
And so how do you develop a potency assay around that? That is still to this day one of the most challenging. You can overexpress the AAVR and think, oh, well, the AAVR, but no, people have overexpressed the AAVR and the virus still doesn't affect cells. So
Interesting.
how does AAV9 being one of the best in vivo viruses just does wonderful. Almost so many of the clinical programs are AAV9 based, but it doesn't affect cells.
Yeah.
So how do you even measure what the heck your GOI is doing?
All right, we'll have to brainstorm that someday in a future session.
Yes, we do.
Thank you very much, Brian.
Thank you.
This has been a complete pleasure, as usual. And
I have this guy to thank for it. Tell me everything I know.
Don't tell them that. Thanks, everybody. Hope this works out, and we'll see you on the flip side.